Welcome to Section 5 of Metabolism. In this section, we'll be discussing the electron transport chain, or ETC. Let's get started. The electron transport chain is a group of complexes which utilize the energy from NADH and FADH2 to produce ATP. This occurs exclusively within the mitochondria. It's also important to know that the proteins of the electron transport chain are produced from the mitochondrial genome. There are two types of phosphorylation which are both used to produce ATP. Substrate-level phosphorylation produces ATP through an enzymatic reaction that adds a phosphate group to ADP. For example, the conversion of 1,3-bisphosphoglycerate to 3-phosphoglycerate is catalyzed by an enzyme that produces ATP. So the enzyme that catalyzes the reaction transfers a phosphate group to form ATP. So ADP becomes ATP. Oxidative phosphorylation refers to the process that generates ATP through the enzyme ATP synthase as hydrogen ions move down their concentration gradient. I'll explain this more in a minute. In this lecture, we'll be focusing on oxidative phosphorylation, which is made possible by the electron transport chain. This is a detailed overview of the electron transport chain, which can be found in Section 5 of Metabolism. Notice from the figure that there are several compounds, such as complex 1 and complex 2, etc., that are embedded in the inner mitochondrial membrane. Also note the various poisons that are shown in red right here, which inhibit these complexes, which we'll talk more about in a minute. As we've discussed in prior videos, a lot of energy and effort is used to generate NADH and FADH2. This energy is utilized as electrons are transferred from these molecules to the electron transport chain complexes. As the electrons move from one complex to another, energy is used to pump hydrogen ions from the mitochondrial matrix into the intermembrane space. So you can see that hydrogen ions are pumped this direction, from the matrix to the intermembrane space. Notice that this results in the formation of a hydrogen ion concentration gradient. You can see several hydrogen ions out here, and you can't really see any in the mitochondrial matrix down here. The hydrogen ions can then be funneled through ATP synthase, which you can see right here. And as they do this, they go down their concentration gradient, and in the process, ATP is formed. So if we remove the blue circle, you can see that ATP is produced right here. As I mentioned earlier, this process I just described is referred to as oxidative phosphorylation. Very briefly, notice that electrons move from complex 1 to coenzyme Q, then to complex 3, then to cytochrome C, and finally complex 4. Also notice that electrons starting from complex 1 do not move in numerical order because complex 2 is skipped. On the other hand, the electrons that start with complex 2, which you can see right below it, do move in numerical order. So you can see 2, 3, 4. Okay, let's move on and discuss some of the poisons that can inhibit the electron transport chain. Rotenone, which you can see right here, is an insecticide that binds complex 1 and prevents electron transfer. Some students use the last half of the word rotenone, so you can see 1 in rotenone, to remind them that rotenone inhibits complex 1. Oligomycin, which you can see right here, is a macrolide produced by bacteria that inhibits ATP synthase. Aspirin, 2,3-dinitrophenol, and thermogenin, which you can see right here, are all uncoupling agents, which means they increase the permeability of the membrane to hydrogen ions. Recall that thermogenin, which you can see right here, is a compound that's present in brown fat. So all of these poisons essentially prevent the electron transport chain from creating ATP and instead result in the production of heat. Two of the more important and clinically relevant poisons are cyanide and carbon monoxide, which you can see right here. These both inhibit complex 4. Complex 4 of the electron transport chain is also called cytochrome C oxidase. Let's talk more about cytochromes, and then we'll return to complex 4. Recall that cytochromes are a class of proteins which contain heme groups. Heme consists of a porphyrin ring with a center molecule of iron. So if we were to draw a circular ring, iron would be in the center like this. Hemoglobin in red blood cells and cytochromes in the electron transport chain are both examples of molecules containing heme. For step 1, you need to know that the iron in hemoglobin is primarily in the 2+ state, so Fe2+ for hemoglobin. And you also need to know that iron in cytochromes can vary between the 2+ and the 3+ state. This ability of cytochromes is very useful because it allows the oxidation state to change, which facilitates the movement of electrons. Okay, with this background, let's return to complex 4. You need to know that cyanide has a high affinity for iron in the 3+ state. Because the iron in hemoglobin is primarily in the 2+ state, and iron in cytochrome C oxidase oscillates between the 2+ and the 3+ state, 2+ and 3+, cyanide only binds to cytochrome C oxidase, and it does not bind to hemoglobin in red blood cells. So again, hemoglobin is in the 2+ state, and complex 4 oscillates between the 2+ and the 3+ state, which I've shown down here. And because cyanide inhibits the 3+ state, this poison is specific for complex 4 of the electron transport chain. On the other hand, carbon monoxide, which you can see right here, preferentially binds iron in the 2+ state. This means carbon monoxide can bind both hemoglobin and cytochrome C oxidase. So carbon monoxide can bind hemoglobin and complex 4. Okay, let's solidify this information with a couple of questions. A 45-year-old male with a history of hypertension presents to the ED with a blood pressure of 190/123 and proteinuria. He is started on a medication used to control his blood pressure, but then develops reddish skin discoloration and a severe headache. Repeat labs reveal a lactic acidosis. This patient's symptoms are most likely due to a substance which has a high affinity for what compound? From the question stem, you should have noticed that this patient was treated with a medication that resulted in cyanide poisoning. Symptoms of cyanide poisoning include reddish skin discoloration, headaches, and confusion. Recall from pharmacology that nitroprusside is an antihypertensive medication that can be used to treat hypertensive emergency. Also recall that nitroprusside can cause cyanide toxicity because the drug contains cyanide side groups in its molecular structure. So let's draw cyanide side groups in its structure right here. As we just discussed, cyanide has a high affinity for the iron in the 3+ state, which means it preferentially binds cytochrome C oxidase in the electron transport chain. This halts aerobic metabolism and promotes the formation of lactate via anaerobic metabolism. This explains the patient's development of a lactic acidosis. So nitroprusside results in cyanide poisoning, which preferentially binds the iron in the 3+ state, which inhibits complex 4 of the electron transport chain, resulting in a lactic acidosis. So this patient's symptoms are most likely due to cyanide, which has a high affinity for the iron in complex 4 of the electron transport chain. Okay, let's do another question. The patient is treated with a compound that results in resolution of his symptoms. However, while drawing blood for repeat labs, his blood now appears brown. What is the mechanism which explains the discoloration of the blood? In order to get this question right, you need to recall that the treatment for cyanide poisoning is nitrite. Inhaled amylyl nitrate oxidizes hemoglobin from the 2+ state to the 3+ state. So we're talking about hemoglobin in red blood cells, and nitrite oxidizes this from the 2+ state to the 3+ state. You should know that when hemoglobin is oxidized in the 3+ state, it's called methemoglobin. Because cyanide has a high affinity for the 3+ state, it will bind to methemoglobin. So we'll draw methemoglobin binding to molecules of cyanide. In this way, the red blood cells travel throughout the body and act as a sponge by soaking up the cyanide. This releases much of the cyanide from the cytochrome C oxidase, allowing aerobic metabolism to continue. So let's imagine that complex 4 is bound to cyanide, and then when the drug is given, the methemoglobin binds up the cyanide, allowing complex 4 to be liberated. The brown-colored blood seen in this patient is a result of methemoglobin. For step 1, you need to know that methemoglobinemia can cause chocolate-colored blood.